Hello, ISMB. So today I'm going to talk with you about large language models and how I think they can help with bioinformatics. But specifically, the big question of where does biomedical or biological knowledge actually come from? We can think of it as the result of repeated observations, but really that's kind of a task in itself to learn from those observations and to automate that process. So how do we actually automate learning from literature, comparing findings, or even just integrating observations across studies, knowledge bases, and across entire fields and disciplines? So if the past few decades have taught us anything, it's that we need structured data. We need consistent data models, standards, and ontologies that can help with this process and help make our data more manageable and more machine operable. But they don't do the hard work of actually structuring the data for us. So how do we curate structured data from unstructured text? Traditionally, this is the domain of human experts, which is a famously limited resource, particularly in terms of time. But we've also developed role-based extractors like the SEMEDDB project that offer fairly high precision with low recall, unfortunately, that can vary by domain and structure that you're actually trying to extract from data. Then we also have methods that are based on term enrichment or annotation enrichment, where you're trying to find some kind of a correlation between the frequency of a particular term and some other factor, like a context or a topic. This unfortunately can sometimes be subject to publication bias, where some things are just studied more often. If you're looking at something like TP53, that's certainly going to show up a lot more than some yet uncharacterized gene. Uh, More recently, there have been a number of applications of neural networks like LSTMs in natural language processing, and specifically this problem of biomedical information extraction. But they have often required really extensive labeled training data. And even then, the kind of data that you're using may actually be overfit for your particular problem and may not represent the broader variety available in biology and biomedicine. So most recently, we've seen the rise of language models like BERT and GPT, which I'll talk more about shortly, which bypass the problem of learning about language from scratch. You start with something that is trained on a broad collection of language and essentially understands what language should look like in practice. Now, many of these methods may still be very effective for some use cases, but I'm going to focus on the use case of how do we translate unstructured scientific text directly into an arbitrary knowledge schema. So these schemas might be complex and involve nested subclasses, and you may have some classes that are one particular type and other classes that are other types, and then you want to know the relationships between them. So it gets to be pretty complex. And then let's take that a step further and say we want to link to external unique identifiers. For example, we want all the proteins to link back to UniProt. We want all of the taxons to link back to the NCBI taxonomy database. So can large language models like OpenAI's GPT-3 or GPT-4, can they actually help with this task? Uh, well, there's a few things we need to consider before we begin answering that question. And one is this concept that bigger models may not always be better. It's really task dependent. And in fact, if we look at recent work with the Big Bench language technology benchmark, they found that about a third of the tasks in that benchmark, and there's more than 200 of them, uh, actually seems to be subject to the emergent behavior of large language models, where they just suddenly get better at some tasks when they reach a certain size. And that's a really valuable thing to be able to take advantage of with large models, because it's not like they're just always steadily increasing in performance as they get bigger. It's like they really do hit a point where they're just capable of new things. And that's an interesting thing to be able to explore. Unfortunately, for many of these models, the GPT-1s included, we're not entirely clear what the training data is. It may even include some kind of fictitious claims that we want to try to avoid. When we couple this with the idea that we are actually getting human-like performance on some tasks, even in biomedical applications, um, we need to be aware that this human-like performance is not actually coupled with any kind of human reasoning. Uh, and to make that a little bit more complicated, our ability as humans to distinguish between human communication and the generated text that we get out of those models is sorely lacking. In many cases, it looks exactly the same. For example, we look at this study by Levine et al., where they were actually trying to ascertain how good GPT-3 was at doing medical triage based on case descriptions. And when they talked to it about vaccines in one instance, it came back with vaccines can cause death. Vaccines are not tested for safety or effectiveness, which is patently false. And an unfortunate thing to have a model try to present you with or maybe convince you of. So this leads to this other complication of hallucinations, where we need to remember that large language models are grounded in language, not fact. They are perfectly capable of inventing things that are patently false and trying to convince you that they, in fact, are true. So to counter some of these issues, we developed this method called SPIRES, or Structured Prompt Interrogation and Recursive Extraction of Semantics. In short, that's information extraction that's grounded in reality. So in SPIRES, you start with some kind of text that you want to extract information from, and you couple it with a knowledge schema. And again, this can be one of these complex schemas with nested subclasses where you actually want to link concepts back to some kind of external identifier. You pass both of these as part of a structured prompt to a large language model, in this case, GPT-3 or 4. You parse the resulting response, ground map, and normalize those with the ontology access kit, which is publicly available. And then you get structured data out the other end that ideally has all the capability of addressing your use cases. So this is currently available through the OntoGPT package, and if you scan that QR code over there, then you can find the program itself. So in the broader sense, this means that structured data can be extracted en masse from large collections of unstructured text, translated into relationships that you would be able to integrate into knowledge graphs. Luckily, we also have some infrastructure to do that as part of the KG Hub project. So see kghub.org for more details about that. So you can combine this structured, extracted sets of relationships with things like ontologies that will be able to better contextualize those kind of relationships into a heterogeneous knowledge graph. So to get a little bit more specific, what SPIRES actually does is it works with a LinkML schema. And LinkML allows you to use a pre-structured language to define the kinds of things that you expect to model in that schema. So what SPIRES does is it generates a prompt from that schema and the input text. You get that prompt, a GPT or your other language model does the prompt completion. You get the completed prompt back from that we essentially call the raw completion. The individual text fragments in there are searched to see if they can be grounded to the specific chosen annotators that we call them. Where possible, we can also translate the result to OWL and use that to do reasoning, if you're familiar with the kind of reasoning that you can do over a nice ontology. So this is a quick example of what a LinkML schema actually looks like in practice before we pass it. Obviously, this is the graphical representation rather than the text. But in this case, you want to define a kind of document that contains relationships between chemicals and disease. So in turn, those documents can be defined by publication metadata. Those specific relationships between chemicals and diseases can be defined based on a subject, object, and a predicate, where the subject is chemical, object is disease, and the predicate is some kind of relationship between them. So I'm gonna step away from chemicals for a second and talk about recipes, because it turns out that this is a great example for doing information extraction. So if you just go online and find a nice recipe for Lyonnaise potatoes, then you can point OntoGPT with the SPIRES method directly at this, and you get a result very much like what you have on the right here. Now, this is actually a truncation of the total result that you get, but you can see right away that based on our data schema that we provided, you get lists of ingredients coupled with the amounts. Now, each of those ingredients are defined based on the food ontology, or food on, so they each have those nice unique IDs. The amounts are based on a given value if provided, as well as a unit from the units ontology, that's UO. So essentially you have a completely structured ingredient list based on values. And in some cases, the language model has had to do a little bit of inference and say things like, okay, well, the state of this item is chopped, or in one of those cases, well, we actually have 4 onions. Onions aren't really a unit, but our language model is essentially intelligent enough to figure that out. Now, we can also go in and, based on the same model, try to say, okay, well, here's the set of actions that needs to happen in the order that they need to happen. So we take those food ontology IDs and say, okay, well, in this case, we need to caramelize the onions, or we need to use our skillet in, well, a particular large oven-proof skillet. Now, in this case, we've categorized that as a food, unfortunately, so there are mistakes to be made sometimes. But some of this really just comes down to lack of a proper ontology for this particular thing. So if anyone wants to start a cooking actions ontology, then I'd be very excited to see it. Now, of course, this works for biomedical documents as well. In this case, this is a study about small cell lung cancer and therapies thereof. If we apply our chemical disease data schema to it, then right away we can extract, okay, we have a particular set of actually 2 drugs that we know are used to treat a disease, and we can look at that disease name back to a mesh ID. In practice, if we actually go through the Biocreative 5 CDR set, that's chemical to disease relations, and see how well it does, it will perform just slightly worse than the average F1 score across all the participants in that initial challenge. The big plus being that, well, this method doesn't require any additional training or fine tuning. It's all in the language model already, and that's pretty convenient to have. Now, does this work on more biological or ecological papers as well? And the short answer is absolutely. So in this case, we have a paper about cellulose patterning in seeds, and our particular plant of interest here is Arabidopsis as a very common model organism, and so we can extract that as part of our model, but it also extracts the genes, the relationships between genes and organisms in case you have multiple different organisms in a given input text. But then it can go in and extract things like the particular activities, gene functions, and cellular processes. And in this case, it's aligning them to Go terms wherever it can. In some cases, we don't have a matching Go term, and sometimes that highlights, well, maybe that's something that Go is just lacking, or maybe this is just a very complicated kind of activity that, again, our LLM is capable of extracting for us, and maybe we're still interested in. And so it goes directly into those extracted bits. So I'm not going to spend too long on this, but if you want to go ahead and try it yourself, now this is the recorded version, so feel free to pause your presentation here and click on that link or scan that QR code, and you yourself can go and try Spires through the OntoGPT package. Now, OntoGPT will happily download all the necessary ontologies that are defined in a given data schema, and you can even create your own data schemas for your own use cases. So I'll close out by mentioning we do have a preprint out on this that's readable. We have a with the full description of the Spires method. Next steps on this are to essentially automate the process of knowledge graph assembly. We want to be able to extract relations directly from the literature, integrate them with those defined in other knowledge bases and ontologies, and essentially use those to identify predictive relationships. This is great because you get the best of both worlds. You get the unstructured, essentially more noisy relationships that you'd find in biological and biomedical text, along with the very structured relationships and the curation that you get from knowledge bases and ontologies. And you can learn from both of them, which is great both if you want to be able to do additional curation in those knowledge bases or in the ontologies, but also if you're trying to combine a whole set or an entire field's worth of literature and kind of contextualize the findings that they get. This is great if you're working across domains as well, because in a particular domain, a certain concept or even a gene or protein name may be referred to using a completely different set of language in one domain versus another. So the language model and the grounding can help to do a bit of the disambiguation and find some consistency across domains and across literature. Now, there are still some limitations of using this method. For now, we are largely dependent on the GPT models available through OpenAI, and we directly use their API. That doesn't work for everything. We don't necessarily always want to send all of our information directly to a foreign API, to a small subset of models with training data that we're not entirely clear on. And we also want to avoid potential hallucinations in there. So what we're doing now is adding more ability to use open models that we can have greater control over and greater knowledge of the training data that went into them. We're also want to try to improve some of the mappings that we get between identifiers. And we've added some additional functionality in a method that we call spin doctor for gene enrichment analysis in here. So the overall goal here is to do some broad literature extraction. In some cases, this can be from full text, like from PubMed Central full text.
Share my one. That one share my one.
So with that, I'd like to say thank you for you, the audience, for everyone in my team in the Berkeley Bioinformatics Open Source Projects group at the Lawrence Berkeley Lab, as well as everyone on the team for the Monarch Initiative. They're listed here, and apologies if I've missed anyone. My email can be found here, along with my GitHub. I'm on Mastodon at the Fedi Science instance. And again, please scan that QR code if you'd like to try out AutoGPT and Spires for yourself.